Thank you, Adira. I think it must be good evening for you now. But anyway, good afternoon, good evening, good morning, whatever, like, because there are participants from different parts of the world. So it's likely different time zones. So I'll be giving a lecture on marine biodiversity. Initially, I'll give few slides on why do you need to study the oceans or the marine biology, and what are the tools to study, what actually the support systems you need, the logistics you need, the instrumentation you need, and then finally we will see the biodiversity and its various uses to the humanity. And I'll sum up the threats to marine biodiversity, which is very, very concerning, and what natural and anthropogenic factors which, posing a challenge or a threat to the marine biodiversity. So this is, in a nutshell, this is the theme of my lecture. Yeah, okay. Then I'll move on to the next slide. So basically, it is, it is planet ocean. In fact, we have, though we call it planet Earth, it is two-thirds is covered by the oceans, and the major ocean, the Pacific Ocean covers almost 50% of the Earth's area. Then Atlantic, then Indian Ocean, then the smallest one is Arctic. And if you look at here, there's again, earlier days we used to call it as Southern Ocean, but now, Antarctic Ocean, now it is, because all this Pacific, Atlantic, Indian, all mixing here around the Antarctic continent, and we call it as Southern Ocean. So it is more justified if we call it planet ocean. And, why do we need to explore the oceans? One reason, as we all know, it covers two-thirds of the earth, because only issue we have is that we don't have the logistics to go deep into the, ocean, either, the offshore regions and explore. It's a bit difficult logistically. So we know little about it. But the fact is that it covers two-thirds of earth. And then it is highly productive. If you look at, the food which comes to us in terms of various kinds of fish, shellfish, and other several things, simply amazing variety of foods you get from there. So it's highly productive. Then, unlike in the terrestrial, I'll show you that later. Unlike in the terrestrial, the habitat is more or less uniform if you look at. Though you can say rainforest or, these deserts or grasslands, other things. But when you compare with the degree of variation in the ocean habitats, actually ocean has got much more wider range of habitats. And it offers excellent biodiversity. The only thing is we are unable to see. See here, lots of fishes, shellfishes may be hiding just below this water. You hardly see anything. This is being a coral reef or lagoon. Probably you see something, but in most places you will not see anything, just like that. But they offers excellent biodiversity, and it is poorly explored and offers, once it is, if anything which is poorly explored means it offers immense potential for those who want to explore. Here I mentioned as biotechnologists or biologists or other people. See, India government now has a major, investment in deep ocean mission. Just wanted to look at the mineral resources, the other commercially exploitable stuff, then look at deep sea life, and how they live at such enormous pressures and low temperatures, and whether biotechnologies can use those organisms or their genetic capabilities for, novel bioactive substances or novel medicines. So lots of scope. Then Why do we need to explore? I'll just go through a few of these slides, like lots of fishes there. These are the kind of reef fishes. You see the ridges along the tail, they can just simply cut your hand just like that. Because I have seen them, I swam close to them in the Lakshadweep Sea. This is myself standing here. We are in the offshore. I'm taking my students to a coral reef island known as Bangaram, where they do snorkeling and see lots and lots of diversity of life, because coral reef is something like, it is known as rainforest of the oceans, where everything is so plentiful, so colorful, so beautiful. One must visit and experience a coral reef at least once in lifetime. So we go there and we allow the students to explore the reef diversity. Your study still, this is simple walking along the beach itself is a learning experience. This is myself walking along one of the islands, coral islands in the Lakshadweep area. And you see a lot of coral, the dead corals washed ashore. I'm just keeping a few pieces which I found interesting for further exploration in my lab. You can see lots and lots of them. And the people, they are basically poor people or resource limited people. They use these broken corals for constructing their house. This is very, very hard material. They cement them together to build the walls and make their homes. And this is another one of my student. He is now a fisheries scientist. So he is Nashad, just avoiding his PhD. He is studying the planktons, because see, the setup of life is like this, whether it is on earth surface or on the ocean, land surface or in the water. The sun is the ultimate driver or the source of energy. Then on land, it is, you can see the plants, green plants trapping the energy. Then the herbivores, eat the plants and convert them into animal protein. And the food chain is so complex and it goes different directions. And finally, everything will die and come back to nature and get mineralized and being recycled. So here, my student is trying to look at what is the plankton diversity here, which is directly a function of the productivity. I mean, the sunlight converted by phytoplanktons or plant planktons, then he is looking at study zooplanktons. When there are a lot of phytoplankton, there is definitely will be a lot of zooplanktons. And you can maybe the oceans are ocean ranching systems there, open sea ranching systems there. This is a kind of cage culture, food production system in the ocean or in the sea, because they offer vast areas, and this vast area you can use it as food production systems. The cage is suspended on these floats, and you put the feed. The net will be so deep here, and you can, depending on the area, you will be putting a lot of fishes here, and the fishes will grow, and their waste material will be flushed out by the tides and currents, which are normally happening in the ocean or in the coastal waters. So the waste management is not becoming a problem here. You keep them in the impoundments or in this netted enclosure, you feed them, you grow them for nearly 6 to 8 months, and you take out a high quality protein meat in terms of the fish. And you can also look at, whether the algal bloom. These are all, we translated all these things into research papers. This is a trichodesmium bloom, which we accidentally saw when I was taking a small country boat to one of those reef islands, those lagoons. Then we could sense this is a kind of algal bloom. Then we actually took a sample of it, then analyzed it, what is causing this bloom, because nothing can happen just like that. So we will look at the nutrient dynamics, because if something has to grow, nutrient has to come, where it might be in the, this morning, Professor Yamashiro from Niigata University was asking me, how, Dr. Hatha, how are you going to connect the geology to the biology? So I said many of the nutrients which are coming from as the rock leachates, the oceans are, I mean the sea bottom is basically rock, and the characteristics are so different that each kind of if it is a rocky shore, it has got a different kind of biota. If it is a sandy shore, it has got a different kind of biota. If it is a muddy shore, it has got a different kind of biota. And you can, I live in an island where I can see how a rocky area is much more populated than a sandy shore. But sandy shore, many things will be digging deep and hiding there. On the rock, many of them will be attaching there. They are very firmly attached. It is very difficult to dislodge them from the storm. So it's very, and many of your marine sediments, those who are in geology, see, you call it as marine ooze, is basically coming, raining from the top. The algal siliceous material, the coccolith ooze, the radiolarian ooze, all those things are basically, biogenic ooze. They are coming and they are forming a major quantity of these sediments in the marine environment. So biology is definitely connected to geology, and the nutrients from the rocks and other things are absorbed by these planktons or shell forming organisms, and store them in their shells or in their tissues, and then they die, they release. So everything is interconnected. And then we can also look at the biodiversity of the coral reefs. As I said, it is one of the most biodiverse area which I have ever seen. And then maybe if you go to the coral reef area, you will see a lot of fishes which are so brightly colored. So brightly colored. The coral reef environment is like it's a riot of color out there. Everything is so beautiful, so colorful. So many of them have got excellent potential to market them as ornamental fish. So the ornamental fish culture, this is a ornamental fish culture facility developed in one of the coral reef islands. And these are two research fellows manning this research station. And this is one of my professor, Dr. A.V. Sarama, who also came with me to the Lakshadweep Islands. So lots you can do there. And see, if you come back to the characteristics of the oceans, the characteristics of the ocean, if you look at, see this, you see this is actually average depth. I'm talking about average depth. There are areas like a Mariana Trench or Mindanao Trench where the Mariana Trench is maybe 11,000 meters plus. The Himalayas, if you put the Himalayas there, then again 2 kilometers water column up above that Himalayas. So, but if you average several points, you take an average depth, it is around 3,800 meters, or you can say 4,000 or 4 kilometers. And the temperatures ranges from -2 to 40 degrees centigrade, because I live in a tropical area. I'm around 9 degree north, 9 degree 58 minutes north, or you can say 10 degree north, northern hemisphere, where summer temperature, water temperature will be around 37. These days it is even going up to 40. And if you go deep, I have gone to Arctic, both Arctic and Antarctic. I have taken sediment samples from the Arctic. Antarctic, I have not done the marine sediment samples. I have done Antarctic lakes, but I've done Arctic ocean sediment samples. The temperature is -2. I've got several readings which were -1, -2 temperatures, right? And this is, if you go deep like this, 1 atmospheric pressure increase with every 10-meter depth. That means you can actually go up to 90 meters without, a kind of, without any device, without any submersible, or you can do a skin diving, maximum up to 90 meters, because that pressure will be around 9 atm. If you go 100 10 atm, and then it is all, break your heart. It is difficult, it is dangerous. Never attempt that kind of depth, skin diving. So it is, you think about depth, I mean, the average depth and the pressure will be around 380 atm. It is such huge. And if you look at Mariana Trench, it is maybe around 1000 atm will be this pressure. So it is, but still, I saw a video very recently in the BBC. Lots of fishes moving there at around 8,000, 9,000 meters. They are lacking pigmentation because there is no point. Pigment is not going to do anything. They're all kind of albinos, or they're kind of white, and they have systems or mechanisms. Their bodies are very pliable, and they have mechanism to withstand such almost 900 atm pressure. So it is really, really huge. So depth wise, it is a totally different scenario there. Then you look at the light penetration. See, last week I was out in the coastal waters for consultancy or related sampling. The light penetration which we recorded was hardly 2 to 3 feet because it was so turbid. It was raining, a lot of land runoff, a lot of storm surge related churning up. So the depth, the light penetration was hardly 2 to 3 feet. I've been to Niigata. I was there in a shadow station where I could record this Sechi depth or the light penetration up to 170 meters plus. Really, so because very clear offshore, there is not much turbid sediments coming into the sea, and this light penetration very, very important, because ultimately your production is basically depend on light penetration. When it is beyond 1000 meters, it is almost pitch black or aphotic zone. You have a euphotic zone, well lighted. You have a dysphotic zone, diffuse lighting, and you have an aphotic zone which is no lighting, where most of the production will be a kind of chemosynthetic bacteria, or those who do some chemical reaction and do the production. And the salinity ranges from seawater basically has got a you call it as constancy of seawater, as well as seas being very huge, voluminous thing. The salinity changes are not that great. Only in the coastal waters, when it is raining, like in monsoon in my place, now it is raining since this morning, it is raining. So where a lot of land runoff, lot of freshwater input takes place, and in such situations, the salinity drops down to nearly, almost like 5 to 10 ppt. Otherwise, it is more or less averaging 3.3 to 3.7%. Then see, these are the biological provinces of the sea. If you look at you could see two different kinds of environments. You see the solid line. This solid line, I am just moving my cursor along this. This is nothing but the sea floor. So this is one habitat. You call it as benthic habitat. So this benthic habitat, lot of organisms living here. They cannot swim. They're simply resting on the floor, and they are living here. There might be detritus feeders. There might be some people here, or the organisms here will be depending on the food, whatever food which is raining from top. Sometimes, as they go deep, there will be very little food availability. So if there is a whale fall, I mean the death of a big whale, it is known as whale fall. It will take at least 1 or 2 years to reach here, but still ultimately the big ponds will reach here and provide some amount of food. So if you look at, this is one kind of a habitat, this is the benthic habitat, and this is the water column here. The entire thing is nothing but water column, right? This water column, so if I take something like this, if I can go, the whole thing is a water column. This is a water column, so this thing is a water column. So where organisms living there are all swimmers or drifters. So this is pelagic environment, this is benthic environment. This is a wave washed area, you call it as a littoral zone. And below the wave washed area, you have a sublittoral zone where the organisms living here will have ability to firmly attach to the rock they have, you know, very strong covering of shell because they are pounded by the waves. So they will have a very strong shell covering, or they'll be shelled mostly. They have a very strong adhesive stuff there, kind of cement like stuff, and they'll be, you know, kind of closing when it is low water or low tide. They're exposed to drying, they're exposed to air. That time they will close their shells very tight and trap whatever water available. They'll trap it inside the shell. So they have a lot of mechanisms which are very different from organisms living below this wave washed zone, which is sublittoral zone. Then as you go deep, you call it as bathyl zone, then abyssal zone, and hadal zone is pitch black and maybe beyond 6,000 meters depth. But if you go to the pelagic, see, this is water column is the pelagic. This area, which is, you know, this area, which is, this is the landmass, this is the continent, land side. So here, this water will always have a lot of nutrients because a lot of rivers draining their material into the coastal oceans. The rains which are bringing a lot of heterogeneous material into the coastal waters. So the neritic waters mostly, or near shore waters, will be nearly more or less nutrient rich, and they will support a good amount of productivity, and that will be reflected into good diversity of fishes, algae and shellfishes, crabs, crustaceans, shrimps, you know, everything will be much more in this region because this is a nutrient rich area where a lot of production happens and it is translated into a lot of diverse forms of life. You go deep, you know, you go from here, you cross the continental shelf margin, then it is basically you are going to the oceanic region. This region is also known as exclusive economic zone, EEZ. In this area where each country has got their own exclusive rights. If, you know, Japan cannot come and do fishing in the EEZ of India, or US cannot go and do the fishing at EEZ of Canadian waters, nothing doing. Or this is an area where exclusive jurisdiction of that particular country, and beyond which it is oceanic, high seas. So high seas, if you look at, this is nutrient poor, this is oligotrophic waters. This doesn't have, only big swimmers will be moving here and there, and they go back here, feed, and they come back here, and like that. So in the oceanic environment, if you look at, because there are a lot of depth-wise classification, you can see the top level of that is up to 200 meters, which is again good productivity there because of the sunlight and some amount of nutrient availability brought here through currents and tides. So this is known as epipelagic. And you 200 to 1000 meters, you call it as mesopelagic. And 1000 meters to 2000 meters, you call it as bathypelagic. And 2000 to 6000 meters, you call it as abyssopelagic. And beyond 600 meters, you call it as hadopelagic, which is pitch black, very little life. But I've seen, as I said, I saw in a week back BBC, there was a wonderful videos which was done by, I think it is Scripps Institution of Oceanography or Woodsole Institution of Oceanography, both from US. So they have used very good underwater lighting to record videos of some kind of fishes which are swimming at great depths around 8,000, 9,000 meters. And then the next question comes, how do we study the oceans? This is a big question. How can we study the oceans? And you know, it is not easy. If I say you go to the 200 meter depth or say 100 meter depth and collect some samples, it's not easy. I can go on land, I can take my car or truck or walk or many means. You can go and collect a sample, but it's not possible. So for studying the oceans, we really require a research ship. This is the most important oceanographic instrument. I traveled in this ship several times. I was you know, the chief scientist in one of the cruises, because each cruise will be, they will have a cruise track along the ocean. This is mostly doing in the Indian Ocean. So they will have a cruise track, and the cruises will be around 14 days to 21 days regular. But maybe sometimes it is a little more than that, sometimes a little less, but mostly between 14 to 21 days. So what they will do, this is known as Fisheries and Oceanographic Research Vessel Sagar Sambada. This is managed by Center for Marine Living Resources and Ecology, that is CMLRE. Center for Marine Living Resources and Ecology. So what do they provide? The researchers provide platform to work, and there are lots of stuff that I'll show you some of them, and they perform multiple tasks. You have an onboard, computer, a lot of automated instruments to look at the depth of the cruise track. You have an AWS looking at the weather vagaries happening in the ocean, and you have space to accommodate many items of equipment. You have, winches to operate the nets for fishing. You have winches to operate the grab samples for taking sediment samples. You have winches for operating these water samplers. And most importantly, they need to be seaworthy. If they are not seaworthy, you cannot take, because the sea is such a huge place where even this ship, I traveled in this ship, when real swell happens, you will be just shaken just like that. You will be really, vomiting a lot. You are really unstable on board, and so seaworthiness is very, very important. And then, I'm just giving you some pictures of my students working, and you can see the huge metal wires rolled onto these winches. This is myself discussing some sampling plan with my students. Several of them are now scientists in various institutions. And this is a couple of students. They are technical officers in Indian Institute of Science, Education, Research, Trivandrum. He is in National Institute of Oceanography scientist there. He is a teaching faculty. So they are actually sieving this sediment to look at what kind of benthic organisms there. So we're all there. So the ship provides a lot of space. You can sit there, you can do your work. You have laboratories within the ship. You have, retiring rooms where you can watch TV, music, and it's a big place. And you also have I traveled in this several times. When I went to Arctic, I went there 3 expeditions. Once I was expedition chief or leader of the Indian Arctic expedition. So this is an ice class vessel, small ice class vessel in the Arctic to collect samples from the Arctic Ocean fjords. You see, this is another instrument which is very interesting. This is known as flip or floating instrument platform. See, this has got a big tube-like thing, long tube-like thing. So this you can fix it anywhere. It is basically floating. So once you reach, you have a GPS, you go to your location, then you start pumping water into this tube, then it starts gaining weight and it starts sinking like this, starts sinking, and finally it becomes upright. When it becomes upright, it provides platform. From the platform, you can actually operate your devices. Mostly this is used for drilling, exploration, oil exploration. They wanted to do test drilling, whether something is there or not, whether oil is there. They will use this floating instrument platform, which you can take it anywhere, and then it stabilizes. Once you pump in water into this, it stabilizes and gives a kind of a nice platform. And as I said, the average depth of the ocean is 3,800 meters or around 4 kilometers. You cannot simply go and take samples by skin diving or anything. So you use submersibles, research submersible. This is one of its kind. This is known as Alvin, which is owned by NOAA, National Oceanic and Atmospheric Administration. I was there in 2013 as a Fulbright scholar. I gave a couple of lectures there regarding the kind of research which we are doing at Cochin University of Science and Technology and the School of Marine Sciences. And another one is my research on microbial diversity in the Arctic as a function of climate change. So this particular instrument is capable of diving up to 6,000 meters. And you see the arms, robotic arms. These robotic arms can actually collect life from here or rocks from here. You might see the geology people may be seeing a lot of things they're collecting from the Mars or the moon using robotic instruments, Mars rover or other things which can actually collect material. You sit here, you operate your robotic arms on the surface of Mars and bring back the material to Earth, and then you actually analyze them. You look for signs of life, you look for what is the elemental nature, you look for organic carbon, several things. So here, this collected deep sea tube worms, this is actually, you might have heard about black smokers. All geology people may be familiar with, plate tectonics and mid-ocean ridges and black smokers. So where the lava comes out, you can see the plumes coming out. So here they collected deep sea worms, and they also, some of those things which are growing at a high temperature, the DNA is highly stable, and we have some fantastic applications in PCR machines or polymerase chain reaction machines. We will see that later. So this is a submersible which can collect samples up to 6,000 meter depth. This is another one, Shingai. This is 6,500 meters. This can dive up to 6,500 meters. This is a Japanese stuff. So major research targets are geography of the sea bottom and the biology of the deep sea. So they will be fitted with excellent lighting systems, underwater lighting, underwater cameras, underwater, collecting, sample collecting arms. So they can study the geography of the sea bottom and biology of the deep sea. Then again, this is another one. If you look at the hyperdolphin, hyperdolphin was completed in 1999. This can go, see, you can see very powerful lighting systems here mounted on top of this one. See, these are all lighting systems. This is a camera. These are lighting systems. So hyperdolphin was completed in 1999. Can dive up to 3,000 meters, and it equips a high sensitive, high vision video cameras, which has 2 sets of manipulators, robotic arms for sampling in the deep sea. So these are all for taking samples from the deep sea. And how do you measure? Like your ship is traveling, how you make the depth measurements. This is a very simple device. Now most of the fishing boats, they have it. What they do is they emit sound from here. The velocity of sound in water is 1,500 meters. Suppose your this is emitted, hitting the bottom and comes back in 2 minutes. That means it is 1,500 meter depth, because 1 minute it takes to travel here, 1 minute to come back. So this is continuously being operated. So sometimes here 2 minutes, here 1 minute, or maybe 1.75 minutes, or whatever, it is. So that continuous transmission, it is an outgoing pulse or a signal from ship travels through water to the ocean bottom, and it is reflected off the bottom and travels back to the ship. So this gives you a kind of imagery like this. Based on that, what is the sea bottom like. Whether there are structures which are actually rising up, whether the structures are kind of soft bottom, whether it is a rocky bottom, whether there are elevations or unevenness, all those things you will know. This is again used as a fish finder. Echo sounder is now being used as a fish finder, because the sound hitting a living layer. Suppose suppose you say there is a fish shoal is here, which is around suppose this is a kind of layer, living layer, and the light is transmitted back from this layer, and this will be different from an abiotic rocky or sediment layer. So they will know there's a fish, swimming, a shoal of fish is swimming somewhere around 200-meter depth or 300-meter depth or maybe 100-meter depth. And then what they will do, they will circle the boat or fishing vessel around it, and they use kind of purse seals to take out the fish. Okay. So here, then another, this is our own students working in this, so they are actually sitting in the cabin in Sagar Samada. They are seeing some of the parameters which are, characteristic of the ocean, the seawater. I was there behind them. I was just, looking at the screen just behind them. I'm there. And then, say again, you wanted to see there are several things. So life is basically, if I am studying life is a function of several physical and chemical parameters. See, without sunlight, nobody can exist because sunlight is the ultimate driver of energy. Or, with a certain kind of salt, you cannot tolerate, or living organisms cannot, or certain kind of pressure, you can't, you have certain rates. So we will see what are those characteristics or features, parameters, which are very, very important. For example, here, they wanted to measure the temperature. The temperature. This is a bathythermograph, and this is like, and you can use another instrument known as CTD. I'll show you conductivity temperature depth meter, which we will see later. So here, there is bathythermograph. It can measure temperature up to 1,500 meters, right? So it is like, there will be a thermal sensor there, and there will be a cable which is attached to the system in the onboard, and you will get it. You can, another way is like use an expendable BT or XBT. You call it as XBT. Basically, what you see here, this researcher is actually shooting an XBT. XBT means once you shoot, it is lost. It is basically traveled down till the bottom, and it has got a spool of wire, and from the sensor, it is connected to the wire, and the wire will be touching this end, it will be touching your computer. So you will get a continuous, real-time monitoring of the temperature profile from the surface to the bottom. If it is 2,000 meters, 2,000. If it is more than that, it is more than that. You get a continuous temperature profile. This is, you use an XBT here, and XBT does like this. Then another thing is, you can see the XBT if you want to look at. This is the thermistor sensor, and this is the prop wire spool, and this is the canister wire spool, and it is basically connected here. Once you shoot it, this will be released. It just travels down and take measurement, and each one will be quite costly, but you will get a real-time continuous measurement till the bottom, right? Another thing, this is a very interesting instrument which most of the oceanographers use it. We also use it because any living organism is connected to non-living entity. On land, you call it as air, water, soil. There it is a different set of parameters. So the CTD these are basically water samplers. These are completely, this is maybe 1, 2, 3, 4, 5, 6, 7, 8. This is maybe around 30 containers here. You can collect, and there are a lot of props here, and the props are supported by a cable. This cable will be coming back to the ship. I'll show you some image. So this is observation signal is transmitted from sensor to the operation room. Sensors are here. You'll go there, and real-time data is collected, and electric power is supplied from vessel to the sensor. You see here, this is myself about to launch, CTD, and this is myself collecting a water sample from, from one of the machines, one of the sampling bottles. See, all these sampling bottles are kept open at the beginning, and it goes down, and this is the sensor cluster, which records temperature, which records depth, which records pressure, salinity, and you can add on sensors. Now there are several sensors. You can look at the chlorophyll. You can look at different parameters. You add the sensors here, and everything is connected through a cable to the ship, to the system on the ship. So here, As they go down, suppose my interest here is, collecting water samples from different depths and look at the microbial diversity at different depths. So I wanted to collect at 50 meter. Once it reaches 50 meter, I'll tap, then 50 meter will close, trapping the water column at 50 meter. It again travels down, I need at 100 meter, I will tap the computer, then this will close. Then at 200 meters, another one, 500 meters, another one, 1000 meters, another one. So I'll trap water samples at the desired depth and bring it back to the lab, and then I analyze the microbes there. So this is actually after hauling up. This will have actually 1, 2, 3, 4, 5, 6. This may be 12 CT, 12 water samplers may be there. This is after collection. Everything it is hauled up and resting there. Now people will come and collect the samples from these water samplers. This is onboard Sagar Sampada. This is myself collecting water samples in a very similar way from the Arctic Ocean. This is myself, and this is my colleagues during my expedition in 2009. Then again, for instrument for biological oceanography, if you wanted to look at, see, this is, I was operating this just a week back in one of the coastal waters. See, this is not a plankton net. You can actually, by adjusting the weight, you can actually float it here. You can float it here. So it will say the water will go through this, and there is a bucket here. There is a fine meshed bucket here. So the organisms go through this. The planktons, planktons are a kind of not active swimmers. They are a kind of drifters. So the drifters will go through the net and get collected in this bucket. And once your operations, or you can even put a meter here, so that you know what is the dia here and what is the velocity of your movement, and then you will see how much water has passed through the net, and you can quantify what exactly the number of zooplanktons collected per liter of your water, and based on that you will see whether your water is very productive or less productive, and several things you can say. You can develop a lot of stories, and once you haul it up, then you detach. This is like, this is a screwed ones. This is just, there is a threading there. You can just loosen it. You take out this bucket and small collecting device. From that you transfer the material. And another issue, how I want to study the sediments, because sediments may be a repository of a lot of pollutants there. Sediments may be a repository of fine fossilized things. Sediments may be a repository of a lot of things. So sediments can always tell you big stories. So how do you collect it? This is, we use a grab sampler. Either this is a small one, Van Veen grab. Peterson grab is slightly bigger one. I'll show you. This is a Peterson grab, and you can see this is operated from Sagar Sampada. A big one is a huge. You need a winch to operate this. So it goes. This is now hauled up. When it is going into the water, it will go like this in the open position. When it hit the sea bottom, this will close. This lock will be unlocked and it will simply close, grabbing the sediment. So if it is a soft sediment like this, it will be full. You will just open this window, the grab window you will open and see how much material is there. This is full. This is actually overflowing. This is more than 100%. But sometimes if your sea bottom is rocky, nothing will be there. If there are heavy currents under the water, then there will not be any material staying there, because the currents are taking away all the soft materials. You may get some pieces of stones and other things. So once you haul it up, you will know what kind of sea bottom is there. So this is a soft, organically rich bottom where the sediment is really, really high. This is Arctic fjords. This is from Konstfjorden. I was there collecting the samples. Then this is again, this is before launch, and this is after collection. Okay, so you see this, going like this, and once it hits the bottom, this will be unlocked and it will grab the sediment there. And another thing, suppose you wanted to study, I said, sea floor also supports some forms of life. So the sea floor is basically keeping a lot of organisms there. So you wanted to look at those organisms. So you can see, use a biological dredge. This is nothing but there's a heavy metal frame, and a net is fitted to a heavy metal frame. Sometimes you can even give electricity to this metal frame. So it will be pulled along the bottom. So the organisms here will be get into this, because it will even chop off the organism, because this metal piece is really, really powerful and blade-like. And also sometimes they pass electricity to it, so it will tickle the organism there. They'll jump slightly and then they get into the net. So this is for collecting samples living on a rocky bottom, and it operates at any depth on a cable or line, generally with a hydraulic winch, and it often rips specimens off and can be destructive. This is my students, both of them. He's a scientist, he's a technical officer, and they are actually looking at samples collected using this one, right? Like these are a kind of brittle stars. You can see a lot of brittle stars there from that biological blood sample. And another thing, this is only certain depth. Maybe you go some 40 to 60 meter and all that. 60, yeah, that's max. So self-contained underwater breathing apparatus. You fit all these things. You have a lighting device with you. You will never do it alone. You will do it with a buddy. There will be a buddy there, and you do it in pairs. I have done this in the Lakshadweep. So this is self-contained underwater breathing apparatus. One ideal thing to study the coral reef biodiversity. The outer reef you can explore, inner reef you can explore, and you by adjusting the gas pressure, you try to sink or you want to come up, right? You can do both, and you have the kind of flippers you use to swim effectively and all these things will be pressure resistant watches, torches and other things, so that it will stay good in below water. This is, you see, there are two scuba divers, two buddies. They are actually doing and exploring. Sometimes they will do the mapping like this. Suppose 1 square meter area, they mark, and then they start looking at whether this kind of corals, how many are there within 1 square meter area, or this kind of corals, how many are there, or some of the fishes, how many are there in such. So this is they will use a kind of data collection by marking the transects and then counting them, right? Now you now understand why do we need to study the oceans, and what are the tools. You definitely need a ship, fisheries and oceanographic research vessel, or any kind of seaworthy ship you require. Then you have seen how you can look at the various biochemical parameters using different tools. Now we look at the marine biodiversity of India. So being a tropical country, all the tropical countries are rich in biodiversity. The very reason is the tropical regions receive a lot of sunlight, and ultimately all forms of life are reflections of sun's light. Everything is a reflection of sun's energy. So when you have an energy gradient which is really, really huge, you definitely have tremendous biodiversity. I moved traveled from tropics to the North Pole, from tropics to the South Pole, and I witnessed the kind of diversity shrinking when you go from the tropics to the polar regions. It is very simple reason is energy gradient in the polar regions is definitely much less. It is more or less permafrost. It is more or less frozen subsoil and cannot support much of life. So the diversity, when you look at the diversity of life, actually we have three levels, major levels of diversity. One is genetic diversity within the species. For example, I'll tell you, we are all Homo sapiens. See, I'm sitting here. You are some people in Japan, some people in Australia, some people in Sri Lanka. See, we are all Homo sapiens. We look different, like, an Australoid and Mongoloid, Caucasoid, and then the other Negroid. All these people look very different, but they're all Homo sapiens. Same genus, same species. See, I am different from my brother or my sister. We are the close ones, but still I'm different. I have certain features which are very, very specific to me. So within the species, there is a diversity. So that is one genetic diversity. Then there are different kinds of species. You might have seen, I showed you some brittle stars, maybe other echinoderms or fishes or elephant or whales or albatross or these Arctic terns, polar bear, different species. Different species, that's the species diversity is huge. Then you also have an ecosystem diversity. Ecosystem means this is a water body. This is a fjord. This is a Norwegian fjord in the Arctic, and this is a landmass here. The organisms living here, totally different. This is a fjord ecosystem. This is a tundra ecosystem, tundra biome. So the tundra biome supports a kind of life different, and this water body supports a different kind of life. Rainforest is totally different from desert, which is totally different from a grassland, which is totally different from a myristica swamp, which is totally different from an alpine system, very different from this tundra biome. So that ecosystem diversity is huge, species diversity is huge, genetic diversity is huge. Put together, you have a great diversity of life. And when it comes to biodiversity of India, India is one among 12 mega biodiversity countries. So 25 hotspots of richest and highly endangered ecoregions of the world are in India. As I said, it is nothing kind of blessing or anything. It is simply we are in the tropical region and the energy gradients are high. So naturally, we are having big biodiversity. It is there in the Amazon. It is all tropical regions. You check, all the biodiversity hotspots will be in the tropics between 30° north and 30° south latitude. It will be having the max. Then biodiversity and human health welfare, whether the biodiversity is important or whether biodiversity anyway help us. Yes, indeed. We must consider our planet to be on loan from our children rather than being gift from our ancestors. So if it is on loan from our children, then you tend to protect it better. Or if you feel it as a gift from ancestors, you plunder it, and finally there is nothing left for the future generations, and which is a big problem these days. And that's why all these field-based courses or field-based investigations are basically to make an awareness among the student community, and they are the change makers. They are the most important tools to make change in a system, in a society. So when the field-based observations, field-based studies are there, they will really understand firsthand that there is something going wrong as a result of anthropogenic influence or anthropogenic, because we are now 8 billion plus. So 8 billion plus, India is the number one now in terms of numbers. So naturally, the natural resources which you use are basically limited. So the way we use it now is not going to be sustainable. So once the people like you are, have an awareness that this need to be protected, this is a very valuable resource, then there is a change. then it is becoming sustainable maybe after a couple of decades. So benefits of species and genetic diversity. Many species that are threatened could potentially provide fibers, crops, and medicines for human use. I put one example, one simple example, which most people know. See, there are many things which you see. You know, what is the difference between a weed and a crop? The simple difference is that a crop you are looking at an economic point of view. So you want to tend it better and make profits out of it. And the weed, you really not interested to study what is there in what are the potentialities of a weed. You are calling it a weed simply because it is competing for nutrients or it is competing for space with your crop. That is your only criterion to say weed. But many times when people started looking at the weed, they found that they are much more attractive than the crop. And many examples are there. I'll show you one example. Then another thing, loss of species also means loss of genes, because each organism has a unique set of genes, and sum total of this offers great diversity. So suppose if you are losing one organism, then you are losing the entire set of something. Suppose you say elephant is extinct. Elephant is extinct means you will never see elephant again. You don't really know why elephant is big. Why elephant is able to make so much bulk of muscle in a short span of time, or you are now said dinosaur is extinct. So what was the genetic capability which make it such a big animal? What was it? You have no clue because it is no more available. It is all extinct. So the collection of genes, you know, it is happening over millions and millions of years of evolution. When one organism is getting extinct, everything which gained over millions and millions of years of evolution, you are losing it in no time. For example, here I put one example from track polymerase from Thermus aquaticus, which is a bacteria living in high temperature waters or thermal springs. And then we know that the DNA is, from this organism is very highly stable. So this is DNA which we use in the case of PCR machines. So you can see this is rosy periwinkle, weed to crop, because this has got very powerful anti-cancer agents or anti-cancer molecules within them. So they started, now they know that this is a fantastic, where many people, thousands of acres in India, people are farming rosy periwinkle. And you can see the ecosystem services offered by the biodiversity. And it encompasses all the processes through which natural ecosystems and the species they contain help sustain human life on Earth. This is lots and lots. If you value it in, if you value it in money, it will be really, really huge. You know, when this COVID was rampant, how much it cost you a kind of oxygen parlor visit? How much it was? You know, the trees are just every moment, you know, whenever the sunlight is available, they're simply diffusing so much oxygen as a byproduct. They don't require it. They just convert CO2 to starch using water and chlorophyll. As a byproduct, they release oxygen, which is so much valuable. You can see here, this one, next slide, you'll see ecosystem services like, you know, biodiversity offers following ecosystem services. Purification of air and water, reduction of severity of droughts and floods, generation and preservation of fertile soils, detoxification and decomposition of waste, and pollination of crops and natural vegetation, dispersal of seeds, still more, you know, cycling of nutrients, control of many agricultural pests by natural enemies, you call it as a biocontrol, you can say, and protection of shorelines from erosion. Like if you have mangroves, we have seen how the mangroves along the east coast of India protected the communities from the powerful tsunami waves, because the mangrove roots are a kind of shock absorber for such a huge amount of energy. I'll show you later. And then protection from UV rays, because you have a nice tree cover or other, which basically takes out the light for them to use, and you get a good shade. Modification of weather extremes, provision of beauty and recreational opportunities, and biodiversity in the oceans. We hardly know, I mean, anything. We know very, very little. The reasons which I said earlier, because it is not easy for you to, study it because of the logistic difficulties. It is, it's a very expensive affair. If I just take a boat for one sampling, I have to shell out at least 50,000 rupees. Just, few hours of sampling from the coastal waters, very close to my lab. So OBIS is an ocean biographical information system. So OBIS is Ocean Biographical Information System. According to OBIS, there are 40,000 species worldwide, and information from extreme habitats will take time, because you have a difficulty in reaching there. You have difficulty in cultivating them, because you need to, develop situations or provide conditions which mimic such extreme habitats to grow the life from there. And regions with rapid loss of habitat and decline in water quality could seriously affect species diversity. And especially seas around the developing nations are very vulnerable. As I said, the population pressure on infrastructure. If you look at Southeast Asia, India, Pakistan, Bangladesh, China, Indonesia, Malaysia, we are teeming with people, and the resources, the natural resources are limited or finite resources. So basically, the population pressure on such resources and the infrastructure is really huge. So there is very serious deterioration of this environment is happening in such places, which is really a matter of high concern, because it's just one Earth. There's no planet B. Everybody is connected. Suppose your tropical seas goes out of fish, or like an unsustainable kind of thing, it is definitely going to affect the advanced economies, because the food and the feed movements are such huge on a global scale. And marine environment in India is, we have a coastline of around 8,000 kilometers, and the exclusive economic zone of around 2.02 million square kilometers. And we have a wide range of coastal ecosystems, such as estuaries, lagoons, backwaters, mangroves, salt marshes, rocky coasts. I'll show each of them, sandy stretches and coral reefs. There are 14 major, 44 medium and numerous minor rivers, which with an approximate total length of 40,000 kilometers. And there is also a remarkable difference between east and west coast. I'll show you an image here. You see here, this is west coast. This is where we stay. My place is here. You see this. This is having a continental shelf which is slightly broader, gently sloping, and this is the east coast. The east coast continental shelf is a little narrow. It is not that broad as this pale light color blue is the continental shelf, and this is here, it is narrow. And I've been here. I've seen the situations here where merging of east coast and west coast. You could see very interesting phenomenon. I mean, I went there 2 years back. See, this west coast is just quite placid waters. The water is here, it is placid. Then water, the wave action on this side of the coast is very, very big, huge, and here nothing. Very, very, very interesting scenario. Here, heavy surf and rocky shores, this one, and this side is different. And the shelving with the beaches, deltas, and lagoons. Here it is big waves. Here it is, a kind of placid waters. And you can see some of them like coastal ecosystems like estuary, which is a merging place between freshwater and marine systems. Estuaries are highly productive because it's a kind of ecotone. In biology, we call it as ecotone because there will be organisms coming from the sea, there will be organisms coming from the freshwater. So the combined biodiversity here will be really much more than in the sea or in the fresh water. And the nutrients, it will be more or less eutrophic. A lot of pollutants will be there. Pollution is a characteristic feature of the estuary. At the same time, highly productive and very diverse ecosystem. And you can see backwater. This is where these are. I mean, my place is like this, where a lot of placid still waters are there. I mean, in around Cochin, there is a lot of backwaters there, which is a big tourism attraction. A lot of people operate houseboats. A lot of people. Currently, I have 3 Japanese professors with me. Hopefully, we will explore some of these areas maybe tomorrow. And then you can see here lagoons, which are actually shallow enclosed waters, mostly in a coral reef area, which are like a kind of emerald blue waters, very transparent. You can see the fishes and other things very nicely and highly productive. And you can see here the mangrove. The mangrove, you see the root system. The root system is like a networking. It's a kind of reinforced root system. And it can break away huge waves just like that without getting damaged by themselves. So mangroves also offers a very nice habitat for fish larvae to live in between this root, enmeshed root systems. And because larger fishes cannot go there and feed them, so they give a kind of protection. They give a kind of food availability, and around the mangrove region, they actually supports a good number of fish stock. Then you can see another salt marshes. Salt marshes are like kidneys of earth, because whatever material coming from, whatever pollutants coming from the terrestrial or land, it could be agricultural fertilizers, pesticides, and other pollutants from the land, which will be moving through this salt marsh and finally into the water body. So they will be held up here. They will be held up, they will be degraded, detoxified, and the salt marshes act like, nature's kidneys. And then the rocky shore, as I said, these rocks provide a different kind of habitat than the other places, and there are lots of organisms, mostly the bivalves and other things. They will be living here. A lot of marine algae you can see, a lot of crabs you can see. You can see certain fishes belonging to Gobiidae, which suckers there. You can they stuck to their these stones and boulders, and you will see a lot of shellfish, mostly molluscans, plenty of them, and crustaceans. And this is sandy shore, where you can see, it is totally different from this one. I have my beach 1 kilometer. If it is like that, the next 1 kilometer may be different like this. With the sea is very huge, so it is resting on the seawall, which provides a rocky substratum. And the coral reef, as I said, it is everything is colorful. It is beautiful, very biodiverse environment. They are rainforests of the sea. And marine environment of India, like west coast, you have heavy surf and rocky shores. East coast is shelving with the beaches, deltas, lagoons, and marshes. And west coast region is of intense upwelling. Maybe geology people may be also knowing upwelling is nothing but cold nutrient-rich waters coming up to the surface. So once the nutrient-rich water is coming up to the surface, there's a lot of sunlight is available on the surface, and they convert the phytoplankton or plant plankton. They convert those nutrients and light into production, into green material or biomass, and that biomass will be eaten by zooplanktons and then fishes, and like that it goes. So here, northeast coast, it is weak upwelling and marked differences in hydrography and ecology. So this is again, I'm just showing that same image here. These, I am not going to take much of your time into this. This is very actual research-based statistics of how much things are there each type. For example, this is showing the characteristics of the west coast and east coast. So this is temperature range, salinity range, rainfall, tides, oxygen, and nutrient. and the other things here. States, territories, union territories, the area, and all those things. These are just statistics. So this is different kind of marine organisms, flora and fauna, and see them here. Then you can see some of the major forms like kelp forest. You can see this is big ones, like fronds are really big, like plantain leaves you can see, and you can see the light is diffusing from top into this kelp forest. And this is seagrasses. You will see them much more in the I mean Lakshadweep area, especially in the Kadamat Island. There is a good seagrass bed there. They have a lot of associated fauna and flora. And this is sargassum. See, this is sargassum. Both are sargassum seaweeds. Seaweed diversity, if you look at, there is a huge diversity in the east coast as well as in the west coast. And you can see sponges, there is a good diversity of sponges on the east and west coast. The 451 species and hard corals. You see, this is the sponges and these are the hard corals. I see lots of them in Lakshadweep. There are also sponges also there in Lakshadweep, plenty of them in the coral reef ecosystem. And these are the kind of crustaceans, around 3,000 species available. And you can see these are having a lot of commercial value, like lobsters, edible crabs, shrimps, again crayfishes. These are all actually high value protein, edible organisms. And you can see molluscans, again plenty of molluscans, around 3,500 molluscan species. Then echinoderms, these are the brittle star, the sea star, and sea lilies, crinoidians, and sea urchins, sea cucumber. Sea cucumber is having high commercial value, but you cannot now exploit them because they are endangered species. And molluscans, plenty of molluscans there available in the Indian Ocean. And major threats to biodiversity are like habitat destruction, introduced species, then over exploitation, because most of the ecosystems, marine ecosystems, large marine ecosystems, you call it as LME, they're all overfished or overexploited and dwindling. Now it is mostly the culture fishery, aquaculture is actually filling the gap because the capture-based fishery is in decline all over the world. Then disruption of interaction networks. So you can see this is a tilapia, a very highly invasive species, invaded the waters in many of the countries. An African, native by African, and they have high tolerance to salinity and other conditions, so they outcompete the native fauna. And this is another African catfish which eats almost everything and converts into fish meat. It lives in a kind of slush-like water. I've seen that. It will simply, voracious feeder. It again, if they released into the, by natural calamity, if the impoundment is broken and if they get into the natural water, they simply finish this stuff in no time. And threats to coastal and marine biodiversity, marine resources are like major source of food for local inhabitants. This is of a major economic value. From the commercial exploitation, annual value is around 7,000 crores. That means 70 billion, you can say. And then population explosion has resulted in increased and indiscriminate exploitation during the last few decades. Currently, there are no pristine environments. Maybe Andamans is an exception in India. Otherwise, all environment, there is nothing like pristine these days. And the natural threats are like storms, waves, and cyclones. Extreme wave action may kill fauna and flora. They break corals, and they shift large amount of sand, and smother the coral reef, and death due to salinity alterations. Okay, so you can see the impact of this is in Kerala coast. You can see this is southern Kerala, impact of cyclone on coastlines. And the human impacts are like anthropogenic activities like developmental activities, which result in increased runoff and sedimentation, eutrophication due to land runoff, physical impact of maritime activities like dredging, collection, and destructive fishing practices. Then pollution from industries and oil refineries. Sometimes the oil will get broken, and then the oil is uncontrollably released into the marine environment, killing a lot of biota. And then synergistic impact of anthropogenic disturbances. So these are, you can see this is a wave breaker structure which is required at times in order to save the cost, but it will alter the system. And you can see this coastal development in Florida, USA. You can see I visited this place. It is, like most of the beaches are private beaches and then very expensive, and it is huge development. You can see it's a kind of concrete jungle out there. So human impacts, fishing using small mesh-sized nets, resulting capture of small fishes. So they have not reached reproductive size. So naturally, once you take out them from the environment, there will not be any replaceable stock left behind. So the fishing go in decline, or the stocks will dwindle very seriously. So now they are using some minimum legal size for the mesh in order to avoid catching smaller ones, but still many small fishes get entangled with the large ones and they will die. So targeted fishing results in generation of trash and non-targeted fish. A lot of bycatch fishes. I'll show you this one. See, these birds are basically eating the bycatch fishes from the thrown out from this trawler. The trawler is capturing fish. Their interest is probably if it is tuna or if it is something else. They are interested in that only. But a lot of things coming into their net, and whatever the economic value they take out and remaining is thrown out. But the thing is that everything has got a role in the food chain. So when such things are being removed, I mean, in a very uncontrolled manner, the entire system will be doomed. So you can see here the kind of fishing, and the sediment runoff can affect, which can have profound impact on fertilization of exomarine species. And if the water quality goes poor, it can affect the metamorphosis of larval forms. Oil pollution can cause serious mortality and decrease in fecundity. So how to tackle? What is the final objective of our, this kind of field-based lectures and field-based studies which you are planning under Sustainable Earth, which is being organized by Niigata University with the 5 institutions from India, 3 from Australia and 1 from Sri Lanka. This is for the Southeast Asian region. So, and of course, Australia in Asia, Oceania thing. So where, when people have awareness, when the people who are the student community have the right kind of knowledge, they are equipped with the right kind of understanding, they will do a lot to protect the environment, to make the planet ocean or planet Earth sustainable. So how to tackle, considering the vast area and the general lack of resources for enforcement, because you cannot enforce everything. It has to come from people to a great extent. How many policemen you employ? It is not possible. It is impractical. Awareness appears to be the more successful than legislation in controlling the damaging practices to the sea. Legislation will be there, but enforcement will be a little weaker because of lack of resources. Then the awareness is the best tool. So because a responsible knowledge society can contribute a lot to conservation of marine environmental resources. And that is the take-home message, right? And thank you everyone for your time. And I am ready to take questions. I am interested in the ship. Yeah, I'll just check this. Where are those comments, reactions, or chat. There are, oh, this is people there, right? So,
You can take the last question.
which question?
Last one.
Yeah, I'm
That's all.
interested in the ship named Flip. What kind of research is Flip used for? It is basically for oil exploration, because it is a floating instrument platform. You can take it to, you can just tow it to any place in the ocean and just pump in water into that long tube-like thing. Then it start stabilizing vertically, and then then you can use it as a platform to stay and keep working on your exploration activities. So it must be available. You can actually, these oil exploration companies, if you ask them, they will definitely will be knowing. And does India have population of grey nurse sharks? So actually, I really don't know what you mean by grey nurse sharks. There are, variety of sharks are available. Even yesterday, I ate some shark meat, which was very nice. My brother, Carcarius taurus. I really don't know exactly how it looked like. I'm not much aware of the shark species. India must be having, I suppose, but now there was a big issue here because shark fin rays are very high priced commodity. The local fishermen basically taking out sharks and they're just chopping off the fins and then leaving it. Very cruel thing. So, I mean, policemen, they captured some of those fishing boats with, huge amount of shark fins stored in their deck. And now it is being monitored. That practice is banned. So I don't know whether these sharks coming under that, and maybe they are suffering, their population has suffered as a result of this. So I really don't know about this particular species. 43 new message. Then what is this one? Any other? I'll just see here. Yes, please come in. Yeah, I'm just at the fag end of my lecture. It went well. So, Adira, were there any questions there from? Yeah,
No, sir, I think
thanks.
there are.
Are people happy to support the fishing bans, or is it difficult to make such people follow the rules? In India, it is a bit difficult always because all countries which are not having a good income, or maybe you can say developing or poor, the poverty is the first thing to be addressed. Otherwise, they will try to take out whatever is available and support their families or their livelihood. So it is a bit difficult situation, but we are trying our best to, I mean, now see, for the time being, India is under trawl ban. The June-July period, you cannot operate any mechanized boats along the coastal waters of India. The simple reason is that many fishes are now fertilizing, or many fishes are now their breeding season. So not to disturb them, so that you have a better stock, which is scientifically proved. So during that time, government is these days is supporting them with the food and kerosene and things like that. They're supporting the local fishing community.